Hello everyone, my name is Hudzaifah An of Fazasmara, and I'm excited to present my science project for the 2025 International Walisongo Science Competitions. My project for this semi-final stage is the effect of visible light and gibberellin on mung bean or Vigna radiata germination. We have all seen a tiny seed sprout and grow into a plant. But what is really happening inside that seed? What signal from the environment tell it that it's the right time to begin its life? It's a complex conversation between the seed's internal chemistry and the world around it. My project aimed to look closer at this amazing process. Let's head to the lab and explore the science. The main objective for this experiment was to investigate how two key factors, first, the visible light, and the second, the plant hormones, gibberellin or GA3, affect the speed and the success rate of the mung bean seed germination. The experiment began with a 2-hour soaking period to initiate imbibition, the critical first step where the seeds absorb water and awakens from its dormancy. Half the seeds were soaked in the gibberellin solution and the other half in distilled water. Next, I prepared 4 containers with a moistened cotton pad as a growth medium. The soaked seeds were then carefully arranged in their designated container. To ensure consistent treatment, each seed received an additional drop of its assigned solution. The groups were then placed under their experimental condition, A1 and A2 under constant light and B1 and B2 inside a box to ensure complete darkness. For the next 3 days, I observed the seeds at the same time every 24 hours, recording the number of germinated seeds and measuring the length of the new sprout, known as hypocotyls. This is some picture from the first day, 24-hour, 48-hour, 72-hour. And after 72 hours, the final result painted a clear and fascinating pictures. Let's first look at the number of seeds that germinated. The data is striking. The two groups kept in the dark, represented by the gray and yellow lines, germinated much faster and more completely by 48 hours. Both dark groups had achieved 100% germination. In contrast, the group exposed to light struggled. The group treated with gibberellin, shown in blue, leveled off at 70% germination. Most dramatically, the control group in the light, shown in orange, only managed to have 2 seeds, or 20% germinate by the end of the 72-hour period. Now let's examine the growth of the sprouts or hypocotyl length. Here the difference is even more pronounced. The sprout grown in the dark experienced explosive growth, a phenomenon known as etiolation. The dark group with gibberellin reached an average length of the nearly 3.5 centimeters. Meanwhile, the light group sprouts were significantly shorter. The control group in the light barely grew at all, reaching only 0.13 centimeters. However, the light group treated with gibberellin showed significantly more growth, reaching over 1.5 centimeters. This shows that while light suppresses growth, gibberellin helps to counteract that suppression. So, what does this data actually tell us? It reveals a fascinating interplay between light and plant hormones. First, what is the role of gibberellin? Gibberellin is a key hormone that promotes germination by activating enzymes that break down the seed stored food reserve. In our experiment, its effect was most powerful under the stressful condition of constant light. In the dark, where conditions were already ideal, the added gibberellin had only a minimal effect, but in the light, it gave the seeds a significant boost to overcome the inhibition. In the dark, seedlings undergo a process called scotomorphogenesis or ethylation. This is a survival strategy where the plant focuses all its energy on rapid stem elongation to quickly reach sunlight. The extreme elongation we see in our dark-grown groups is primarily driven by a powerful hormone called auxin, which promotes cell elongation but is degraded by light. In the darkness, auxin is stable and works at maximum capacity. At the same time, gibberellin are also highly active, promoting growth by causing the degradation of growth-repressing protein called DELA. Conversely, when exposed to light, the seedlings switch to a program called photomorphogenesis. Light acts as a signal to stop elongating and start preparing for photosynthesis. It does this in two main ways. First, it degrades auxin, which is why the hypocotyls in our light-grown groups are so much shorter. Second, light suppresses the plant's own gibberellin production, causing DELA protein to accumulate and halt the elongation program. The plant then redirects its energy to develop its autotrophic apparatus, sturdy stems, and green leaves for photosynthesis. This framework perfectly explains the effect of the gibberellin we added. In the dark, the seedlings were already at maximum elongation due to high natural auxin levels, so adding extra gibberellin had a minimal effect on length. However, in the light, where natural auxin is degraded and natural gibberellin is suppressed, the seedlings are in a growth-inhibited state. By adding external gibberellin, we provided the growth signal that was missing, allowing group A1 to significantly overcome the suppressive effect of light compared to the control group A2. In conclusion, to answer our initial objective, the development of Vigna radiata is governed by a switch between two programs. Darkness triggers an auxin-driven ethylation program for rapid elongation, while light initiates a photomorphogenesis program for photosynthetic development. The effectiveness of externally applied gibberellin is more pronounced under the inhibitory condition of light, where it helps to overcome the natural suppression of growth. This research highlights the complex, context-dependent nature of plant hormones and provides a clearer understanding of the elegant strategies seeds use to ensure their survival. Thank you for your attention.